Welcome to Mind of a Scientist. It is my great pleasure to introduce Chloe Ghent and Sabrina Mann from the Darisi and Morgan Labs at UCSF. Thank you so much for joining us today.
Thanks for having us.
Yeah, thanks.
I can start by introducing myself. Yeah, so I'm Sabrina, and I currently work in the Darisi lab at UCSF. And I am from Santa Rosa. I grew up there, and I went to school at UC Berkeley, where I met my best friend Chloe. And we both graduated in 2019. And since then, we've been working full-time as research associates in labs at UCSF. And that we kind of just wanted to take some time to work in between figuring out like our next steps. But I'm actually going to be attending medical school starting this year. And I'll let Chloe introduce herself.
Yeah, hi again, I'm Chloe. So I will be starting a PhD program in the fall here at UCSF actually. So yeah, I guess we can talk a little bit more about the specific work that we've been doing the past 2 years in our respective labs. So I'm going to go ahead and share my screen. So I work in the Morgan lab here at UCSF. We're a biochemistry lab, and we focus on questions related to the cell cycle. So the process of cell division, mitosis. I work specifically with the protein involved in chromosome segregation. And so basically my job is to use these proteins in different biochemical assays to be able to evaluate their activity and characterize them better. And so in order to do these assays, we need a large amount of these proteins. And so to express them, we use both bacteria and insect cells. And to express proteins in insect cells, we use what's called the baculovirus expression system. And so basically this expression system exploits viruses. So viruses are dangerous and harmful to us and our cells because they're able to hijack our cells' machinery to produce in mass like their own proteins. And so we've been able to use that to our advantage by using molecular cloning to incorporate genes that encode for the proteins that we want into the viral DNA. So then we're able to infect insect cells with these viruses that are coding for the proteins that we want. And so that the viruses in those insect cells will mass produce those proteins. And so after we harvest those cells that are expressing these proteins, we then need to purify them with a system called the ACTA. And basically the ACTA has a bunch of different protein affinity chromatography columns. And so these columns are basically filled with a bunch of beads that have molecules attached to them that will bind to a specific tag that we've encoded with the protein that we have produced through the baculovirus expression system. So we're able to catch the proteins that we want using their tags on these beads that are in the column. And then we're able to elute them with a competing molecule and an elution buffer that allows us to then extract the protein off of these beads. And so now we have the purified protein that we're then able to include in biochemical assays. And we're able to evaluate the activity of the proteins, how they interact with other proteins. And that just helps us get a better understanding of how the process of cell division works. And this is helpful because it could potentially be informative for trying to understand disease models that are related to proliferative cell growth that is caused by unregulated chromosome segregation. And it also is just helpful in potentially thinking about how we could exploit the processes of the cell and apply that to translational research in medicine. Yeah, so that's basically what I've been working on, kind of the gist of it.
Cool. I'll talk about a bit what I work on. Here we go. Okay, so I'm in the Darisi lab, and the Darisi lab is both a basic science lab and a translational science lab. And so basically that means that for translational stuff, we're doing research that is supposed to directly apply and have impact in a clinical setting and in humans. And so two main arms are the infectious disease research and the autoimmune disease research. And so for infectious disease research, Mypi Jodarisi is famous for like using this approach called metagenomic sequencing. And basically doctors from all over the country and even around the world will kind of send him samples as a last resort because they don't know what's like causing their patient to be ill, because like whatever they run their typical clinical panel of tests in the hospital like isn't showing up, like it's negative for all these things. They don't know what's causing their patient to be sick, so then they can't treat them. And so as a last ditch effort, my PI has become famous for receiving these samples and like trying to figure out and kind of being like this disease detective. And so we'll receive samples. We've received heart tissue, brain tissue, spinal fluid, blood, and a lot of times it's from people who are in the hospital and are critically sick. And what I would do in the lab is I then take these tissue samples or blood samples or spinal fluid samples, and I extract the nucleic acids out of it, so the DNA and the RNA, and then I put it through a series of steps to prep it for sequencing. And basically what we're doing is trying to get a picture of everything that is inside the sample, because normal tests, what they're just looking for one thing, you know what you're going into, like looking for it. Like I want to look for adenovirus, I want to look for influenza, but metagenomic sequencing is better because it's like a broad picture. So we take all of the RNA that's in that sample, and it'll be RNA from like the human themselves, but also from like maybe what's ever causing them to be ill. But there's a lot of other things in there, like kind of like garbage, like that will wash out the signal. So like a lot of people have like E. coli, or like bacteria on their skin, and that'll like not be what's causing them sick, but it'll still show up in our sample. So that's like one disadvantage of metagenomic sequencing, because like it'll show everything, but hopefully what we can see is what's actually causing their illness. So I put the sample through a series of steps to library prep it, and basically it's modifying the ends of the RNA. You're adding on these different adapters, and then eventually able to put on an Illumina sequencer. And an Illumina sequencer works by taking pictures, like the long stretches of the DNA bind to this flow cell, and the Illumina sequencer takes pictures, and it'll like record whether the base is a C or a T or a G. And then with that information, we put that on a bioinformatic pipeline, and it's able to assemble and align the DNA that was read on the Illumina sequencer to all of the known DNA and viral genomes of like everything that we know. And so if it matches certain things, we're able to say, okay, we know that this is in the sample and this is in the sample, but what is it that's maybe causing their illness? And so that's when you kind of have to use like your educated guessing. And so that's metagenomic sequencing basically. And sometimes it works, and then we see like, oh, there's actually this virus, maybe this is what's causing their illness, and this like aligns with the symptoms that they have. But a lot of times, sometimes it doesn't work. And so it's kind of a last ditch effort, but it's a lot of fun, and it's a really great picture into what is actually in that sample. And another thing that the Darisi lab is known for is doing this on a high throughput scale and using robotics. And so that's a fun thing that I got to do in the lab a lot, was using these really cool robot machines to process 384 samples at once. So we can look at 384 patients at one time, which is amazing, especially if they're all have some sort of similar phenotype, because then like the data that we get at the end, we're able to like draw a lot more conclusions if we have more patients in this like study, right? So one robot we use is called the Bravo, and basically it's like a liquid pipetter, and you can see it here. Basically it is able to move around and pipette 384 samples at one time, or 96, you can change the head. And so that's really makes it a lot easier to do that many samples at one time, but it's pretty neat. And then the other robot that is my favorite robot is called the Echo. And basically it uses these sound waves to move liquid without like any disposable pipette tips or any disposable labware. And so it's really neat in that way. And here's a little video demonstration. So it uses these sound waves from the bottom, and the sound waves vibrate the liquid up into a destination plate. And you're able to tell the robot like how much liquid I want moved at one time. If I want like 10 microliters or 50 microliters, and into what well. And it's really cool. And it also has a big advantage because you're not using disposable labware. It's not like generating more waste. Anyway, so that's one of my favorite robots. So that is one arm of the Darisi lab and the research that I've been doing. The other arm is autoimmune research, which, here we go. And so for autoimmune research, we use this technique called phage display, and phage refers to a bacteriophage, and that is a type of virus that is only able to infect bacteria. And the phage that we actually use are these engineered phage that are able to only affect a certain type of E. coli. So when we're handling it, they're not dangerous to us. And the cool thing about phage is it's really easy to edit their genome. And so what we do, or what we've done, is that we edit their genome. So on the head of the phage, which you can see in the diagram on the right, we're able to change what is displayed on the tip of its head. And what we're typically known to do is use this library called the human peptidome. And so what we've done is we put every single peptide or protein that is in the human body on different outside of the head of the capsid of the phage. And so we have this library or this like solution, and it's like this clear liquid, but it's made up of a ton of different phages, and each phage has a different protein on its head. And this is really cool because when we then take these phage and we incubate it with human serum, and a lot of times we're using human serum from people that have autoimmune disorders. And autoimmune disorders are basically like when the body attacks itself. And so a lot of times the body, for some reason, is attacking a certain protein in itself. And so that protein is more proliferate in their serum. And so there's antibodies that are in the serum, and the antibodies will then bind to the phage heads. And then we're able to like pull them out of the rest of the phage, basically using something called immunoprecipitation. And then sequence what that phage was that was being pulled out and figure out, oh, this is the phage that maps to this antibody. This is what is targeting in this autoimmune disorder. So we'll get like basically patient cohorts like lupus or autoimmune hepatitis, a bunch of different disease cohorts. And we're able to see like, is there a commonality, a common autoantigen that is like attacking their immune system? And if so, this can hopefully then translate to an easier clinical diagnostic, because some of these autoimmune disorders, it takes years to be diagnosed, and it's hard to like figure out that these symptoms align with this disorder. And prolonging that diagnosis prolongs their treatment. And so a faster diagnosis would be faster treatment, better standard quality of life. So the whole point of this translational research is to eventually be more applicable, like relate to the hospital. So that's the autoimmune disease research that we do in the Darisi lab. And then most recently with COVID, we've also been doing COVID research. And this is data from a real study that we did in the Mission District in San Francisco. And basically what we wanted to do is have an understanding of like what variants in the mission were present in the Mission District and of these people. Because at the time that we did this paper, there's this a lot of hype and concern about this variant called the West Coast variant. And it's also known as the B1427 or 429. And people were worried that because it started in, I think, Southern California, and it was like spreading really rapidly throughout California. And people were worried, like, is it more dangerous than other variants? Should we be concerned? Does it spread faster? And so basically this study had a site in the Mission District, and they were testing people, whoever wanted to come and get tested. And basically on the lower right is a map of where all the people lived. So even though it was specifically set for the Mission District and like for people who live in that area to get tested, there are still people that came from all around the Bay Area. But it's a good picture, right? Because then we can see like of these people who got tested and what variant they ended up having, this is where they live, and this is where we know it's been. And so what we did is any positive sample, they did nasal swabs, any positive sample was sent to our lab, and then I extracted the DNA and the RNA from the sample, and then I sequenced it and was able to figure out what variant each sample was. And a lot of times it was, any of these different variants that are shown up on the right, on the top, but sometimes it would be this California variant. And so then with household data and other demographics, we were able to see, okay, like of the people who had the B1427, they were this age and this ethnicity, and this is where they lived. And we're also able to see that it rose to, it like increased in prevalence in the sites that were tested. So on the lower left is like the time frame, and the red is the West Coast variant. And you can see that in November, it wasn't that abundant of the positive tests that were in the Mission. There was a lot of other strains that were there, but not so much the West Coast variant. But then you can see it pretty rapidly spread throughout the neighborhood and became one of the most dominant strains that was in that neighborhood. And so by the end of January, it is one of the most prevalent ones there. And this isn't a factor of sampling size, but as you can see, like, yes, the number of tests in the beginning in November wasn't that much, but towards the end of January, we're getting like pretty equal number of tests every day. And so that was a really nice thing, or neat thing to understand, is that the virus, we're getting a better picture, because I think the biggest thing about the pandemic is that we don't know what we don't know. And so any information is really helpful. And then we were able to like correlate, we were able to see like, okay, of these people that had the West Coast variant, with just basically like survey questions for the people who are sick, we're able to see like, okay, like they didn't have this many symptoms, or this many of them are asymptomatic, but a lot of them weren't that much more sick than others. And they didn't have that much more virus in them either, because some people were thinking that they had a higher viral load than the other variants, but we found out that that wasn't true. So that was a really interesting study that we did, and the most recent research we've been doing in the Darisi lab. And also with this study, this same site has been vaccinating people as well. And so everybody that comes and gets vaccinated at this mission site, they try to enroll them in a study to then later test them weekly. So they try to recruit people back to come every week, either with like gift cards, and then basically to swab them. And the whole point of this is to see that if they turn up positive, are they positive because they got a new strain of the virus that is not resistant to the vaccine, or the vaccine isn't able to neutralize? And then we're able to see like, okay, is this a cause for concern? And we pair that with also antibody profiling. So if they, sometimes we'll get a sample and it turns out that the patient was positive for this virus, but they never actually produced antibodies for whatever reason. Maybe they're on some sort of medication, or maybe they were older. So it's important to like know both sides of that picture. And yeah, so that's some of the COVID research we've been doing. Do you want to talk about your COVID research?
Yeah, so briefly, our lab also started on a COVID-related project, kind of towards the beginning of the onset of the pandemic. So last summer, we focused more on trying to understand the way in which the virus functions within the cell. So the virus itself is made up of the RNA and then also the proteins. And so we noticed that different states of this one protein, the N protein, was able to kind of direct different functions of the virus. So we were able to use biochemistry to kind of dissect how the protein was able to adopt different states, and then also how these different states were affecting the function of the virus. So the virus has to be able to transcribe its own RNA and then also repackage it so that it can leave the cell and infect other cells. So we were kind of interested in understanding that, and then that research could potentially lead to like a more translational perspective of trying to see what parts of that process can be intercepted and treat or like prevent the infection of the virus to like proliferate. Yeah, so most of my research is kind of the back end basic science part of research, not as translational. But yeah, we do kind of the basic science, so more translational research can be done to like apply that to cure and prevent disease.
Thank you. Well, I think we're ready to take some questions. So please put your questions in the chat, but could you tell us what were your high school science classes like?
Yeah, so high school biology, AP biology was kind of the class I took where I really fell in love with science and biology specifically. I just love the idea of being able to understand life better and being able to understand how we're able to function kind of at this macroscopic level because of so many intricate microscopic processes that happen. And so being able to further understand that and unravel that has always been really interesting to me. So yeah, I took AP biology in high school. It was quite a few years ago, so I don't know how much of the course has changed, but yeah, I took that. I didn't really have much exposure to things like physics or chemistry in high school. That was more something I learned in college, but I also gained an interest in chemistry in college, and that's kind of what allowed me to want to focus on biochemistry as kind of like a subset of biology. Yeah, that's kind of my story, I guess.
And then my high school was pretty different. It was a Waldorf high school, and so it didn't offer AP classes. And so I don't think that I got necessarily the same like foundational information about like biology. I think I remember getting to college and being very much like, I didn't learn a lot of this stuff, and a lot of my peers had because they'd taken these AP classes. But I do remember from my high school classes that it was big on like actually going out into the field and doing experiments. So we would like go to the creek and like get like pond water and then like go back to the lab and see what's there. And so that was really fun. And I think I like that like translational impact, like what we were doing in the field really related to like the classroom. And so I think that like started my love for like translational science research. But yeah, we didn't have a lot of that foundational stuff that I think normal people do get.
Stefan asks, can the flaw in metagenomic sequencing lead to incorrect diagnostics of illness?
That's a good question. So because, so long answer, short answer is like, if you contaminate the sample, potentially, but we always run a water control. And so we have just pure water alongside it. So we're sequencing everything that's in that water. So anything that's like would lead to incorrect illness would come from like me, like my own contamination, or from the lab or other people's research. And so that stuff would get into the water sample, so would be sequenced as well. So if we see that in like this patient, there's influenza A, but we also see it's in the water, then we're like, okay, well, maybe that's actually not real. Maybe that's just background contamination. But everything that's in that sample should be a real picture of what's in it. So there shouldn't be nothing that would lead to incorrect diagnosis.
Mm-hmm. And Stefan follows up, how close is your autoimmune disease research to being implemented in hospitals?
I would say it's pretty far away. A lot of it is exploratory. And so we'll work with other labs around UCSF and around the country too. And they'll be like, we've been collecting these samples from all of these patients in the hospital, not with the intent of treating them individually, but with hopefully having some impact in the future. So that's a good disclaimer, I guess, is that a lot of this is just to like understand more about the disease, and hopefully one day it can lead to a diagnostic test. But right now, it's just like learning more about it because so much about these diseases is unknown.
Mm-hmm. Rosalie asks, what is your average workday like?
Yeah, so it really varies, which is something that I enjoy about research, is it kind of ebbs and flows in terms of like the hecticness of the day and what type of things I'm doing. So they're kind of different stages of the process. So as I talked about before, we express the proteins, purify them, and then do biochemical assays with them. So it really changes day to day what I'm doing. So I could be doing like a longer protein prep day, which usually takes up a whole day, or things like molecular cloning, like getting the gene the gene for the proteins that we want, getting it into the right plasmid so that we can express it. That takes a few days. And then biochemical assays, I can also do like maybe a few in a day. So it really depends. But I think now that I have a better handle on the different skills and techniques that I need to do all these things, I'm better able to manage my time. So I'm able to kind of design my work day so that I'm not working crazy hours. So I'm able to still work a manageable 8 hours a day, I would say.
Yeah, I think similar. When I was first learning, I was having to shadow other people. And so I wasn't really in control of my schedule. But like now that I know how to do everything, I'm able to try to plan better. And so I know if I have a really long day one day, because I have to like we get a sample from a sick kid and we want to know as soon as possible, like what's in that sample, I will stay like pretty late and like work a 12-hour day. But like to compensate, if I get everything done, maybe I'll like take a shorter day the next day. So it really varies. And another thing is that like a lot of times you'll have like these long incubations in research. And so I'll have like a 2-hour incubation. So I'll have to like think, okay, like in these 2 hours, can I start another experiment, or should I do like computer work, or like what can I do at the same time? And so that's a really fun balance. And the other thing is like at this lab, I had no experience of like how to code or any like data analysis before I joined this lab. But because I didn't take it in college. But when I joined this lab, I realized that like I'll generate all this data, but then like I'm not able to analyze it. I have to give it to somebody else too. So I wanted to like learn how to do that. And so I was able, like during these long incubations or times where I had downtime, I started taking Python classes and R classes. So I was able to like actually analyze my own data. And so I think like towards like from like the later half of my time at this job, I've been able to like balance more like the research side and the data analysis side, and kind of, yeah, use the two in my daily job.
And nowadays we have a lot of students learning Python. Do you think they'll kind of have an edge in the lab?
Oh, totally. I think that's like such a valuable skill, is like if you can learn any coding in high school, like definitely do it. And then especially in college if you can, but in high school that would be, yeah, amazing.
Yeah, I definitely agree. I think coding like universally will be a skill that you can apply to almost any job, especially in biology with like, yeah, data analysis. R is like a super helpful coding language for biology analysis. Yeah, definitely.
Yeah. I think nowadays they're starting to expect that like everybody is able to be able to analyze their own data. Like maybe back then they didn't actually have that expectation that everybody could code, but like it's starting to become more of a thing.
Mm-hmm. I'm curious, who maintains the lab robots like the Echo?
Oh, so the Echo is actually in a communal space. So a bunch of other labs are able to use it at the same time, or not the same time, but like able to use it. So there's this core facility called like the Center for Advanced Technology called the CAT at UCSF. And I think that that facility has people who maintain it. And like if it's down for some reason, but also each user is supposed to be trained to like troubleshoot it and do daily maintenance on as well. So like I always like make sure that it's done its daily checks before I use it and make sure it's actually going to do what I tell it to do.
Cool. Rosalie asks, what are your favorite credible sources for science news?
Yeah, so I think bioRxiv is a decent place to go. It's kind of where people can post unpublished papers that they're working on. So they haven't necessarily been peer reviewed or published in a more respected journal, but you can kind of see like preprints there. And they're usually from credible labs and credible people. It's kind of an easy way to see like what's new and what's out there. I think beyond that, sometimes I'll just go to the lab website of a professor or scientist from a credible institution and look at the papers that they've published there, which I know will be papers in the research area that I'm most interested in. Yeah, so just going directly to a professor's lab website.
Yeah, and about the bioRxiv, that's like a newer thing in the field, I think, is that people are really trying to do, what's it called? Were you like
more accessible?
Yeah, more accessible. Like a lot of journals now are starting to require that people publish it on these bioRxiv, like open source information places at the same time while they simultaneously submit to like a journal for peer review. And the whole point is just like to get information out there, and so it's accessible and yeah, open source. And so while bioRxiv hasn't been peer reviewed, it's a great place, especially for COVID research. It was really good to like, if you want to know what's being done right now, you go to bioRxiv, because the peer review journals will take like weeks or months or even years sometimes.
Cool. Rosalie asks, do you have any additional advice for students interested in going into STEM careers?
Yeah, I think maybe a piece of advice I can give is just to pursue, I guess, what you're interested in. I think that I was always super interested in biology, but I didn't know necessarily going into college what careers I could get pursuing that besides going straight to med school, which is a great option. But I would just say that there are other options. You can continue to do research with like a biology degree. And it's applicable to also other career pathways as well. But I would just, say don't be afraid to pursue like maybe a niche subject in biology that you're super interested, because I think that is like a fruitful path to go down, and you can make a career out of most biological subjects.
Yeah, and I think I would advise students to really try to like explore everything that they can. Like if they think they're interested in this type of research, like just email people and be like, oh hey, like can I shadow you or can I do this internship in your lab? We in the Darisi lab we have like high schoolers and college kids come in and do summer internships all the time, or even during the school year. And I think that's a great way to like get your toes wet and like get a feel for it if you like it or not. And I think the biggest piece of advice I learned is that like people love to mentor and they want to help other like people who are interested in it, right? I think I was always pretty nervous about like, oh, am I a burden or am I bothering them? But I've learned that like PIs and other postdocs and other researchers want to like mentor other people. It's like it gives them a lot of joy a lot of times. And so like don't be afraid to just like cold email people. When I was at Berkeley, I realized at one point I was like, oh, I need to try to get research and like see if I like this. And so I cold emailed like 10 professors, and the ones that got back to me were like, yeah, of course. And so then I just joined a lab, and it was like a lot easier than I thought it would be. So I think just go ahead and like email people.
Yeah.
That's
I think jumping off of that, just it's super important, I think, to get that lab experience. And I think that I would say to not be discouraged maybe by not having the best grades or academically performing super well, because I think the in-lab experience is much more valuable to yourself, to be able to see if you can see yourself in this career doing this every day, but also just continuing down that career pathway, whether you apply to jobs later on or apply to grad school or med school. That in-lab experience is really important to have.
that's great advice. And lastly, what's your favorite part of your job?
I think my favorite part is like working with patient samples, because I know that this came from a real person and like this information could impact them. I think I really enjoy like knowing. It makes it more stressful sometimes. So I'm like, oh my God, like this piece of heart tissue, like it's so precious, or whatever it is. I'm like, it makes it more stressful. I'm like, I have to do everything perfectly and like puts more pressure on me. But I kind of like that stressful like pressure environment. And so and I also like that it means more. And so I think that's probably my favorite part of my job.
Yeah, I think probably my favorite part of my job is kind of the discovery aspect and how kind of it changes day to day, like what I'm working on. And it's just exciting to, in certain moments, feel like if you've maybe discovered something new or a new reaction or result piece of data. It's kind of like you're the only person at that moment who knows that. And that it just feels really exciting to be able to have the freedom to just kind of test out new things, because there's so many things that are unknown in biology and science, and just being able to pursue that and see where it goes and just have that freedom to explore, I think is really cool. Yeah, I think definitely in science there are periods of lows where it feels like everything is failing, but then those moments when something does work out and you do get a really cool piece of data, it just makes it even more exciting and gratuitous. So
Yeah, to follow up on that, like I think, yeah, maybe we don't want to like sugarcoat it too much, but like there are a lot of failed experiments in this. Like a lot of things don't work, and that, like Chloe said, it makes like when it does work so much more exciting. But I think that's an important thing to know about science is like a lot of times it won't work out, and like you have to troubleshoot and figure out why. And so, that's like the real aspect of it.
Yeah, being able to persevere through that is important.
Well, thank you both so much for joining us today. Such a fascinating presentation.